Hey everyone. All right, we are back with Claudia presenting on oxytocin, intricacies, and implications of the elusive neuropeptide. Claudia, take it away.
Wonderful. Okay, wonderful. Thanks, Sophia. Hello, everyone. My name is Klaudia Szczech. I hope everyone is enjoying the conference thus far. I'd like to thank HAZA for allowing me the opportunity to present a recent paper of mine. Today, I'll be talking to you about oxytocin, its effects, potential therapeutic implications, as well as associated challenges. So my presentation is titled Oxytocin as Therapy, Intricacies and Implications of the Elusive Neuropeptide. And this project was supervised by Dr. Arla Good at Ryerson University. So first I will share some background information on oxytocin to get everyone on the same page. That will then lead into the context in which I completed my research paper. I will then go over my methodology and approach, and we will have a look at the results. Finally, we will get into the broader discussion of the material. I will conclude by putting all of this information in a grander scheme, and we will finish up with any questions you may have. I would love to hear any questions that may come up, so please do hold on to those until the end of the presentation, and I'll do my best to fill in any gaps. Now, you may have heard of oxytocin and its effects in your undergraduate courses, say in physiology or in neuroscience. You may also have come across headlines such as this one from Medical News Today. What is the link between love and oxytocin? Or this one from The Telegraph, oxytocin, the love hormone, could cure shyness. Or maybe even this cheeky title from the Indian Journal of Endocrinology and Metabolism, The Orgasmic History of Oxytocin, Love, Lust, and Labor. Regardless of the source, oxytocin has garnered considerable attention from both scholarly and lay communities for decades. So what is it exactly and what does it do? Since its discovery in the early 20th century, oxytocin has been under scientific scrutiny. We now know that it is produced in the hypothalamus, as you can see indicated in the diagram on the right, and it is then released by the posterior pituitary gland. Oxytocin functions as both a hormone, you can see on the left-hand side now, as well as it functions as a neurotransmitter. So this distinction is based on the method through which oxytocin exerts its effects. As a hormone, it travels through the bloodstream and acts on receptors found in various peripheral tissues, such as the uterus, the mammary gland, the kidney, and the heart. Additionally, it functions as a neurotransmitter throughout the brain, with identified action in the prefrontal cortex, the limbic system, specifically the amygdala and hippocampus, as well as other subcortical regions. Now, in terms of function, oxytocin has consistently been implicated in mammalian birth and lactation, the word itself being derived from the Greek words for quick birth. It has a role in facilitating parent-infant bonding and reproductive physiology, hence the love hormone label. Further, oxytocin has been observed to exert an effect on several facets of socioemotional functioning, notably emotion recognition, interpersonal trust, empathy, stress management, and social bonding. Neurologically, oxytocin and its receptors have been linked to what is called the social brain network, which includes prefrontal cortical areas and limbic regions, as mentioned in the previous slide. Also, recent genetic studies have identified single nucleotide polymorphisms in oxytocin pathway genes that are significantly correlated with social deficits and psychiatric disorders. So based on these preliminary findings, researchers have begun to explore the value of oxytocin as a viable and promising therapeutic agent for neurodevelopmental and psychiatric disorders defined by social impairment, such as autism spectrum disorder, schizophrenia, and social anxiety disorder. So intrigued by its role in social bonding and as a potential therapy for those suffering from socio-emotional challenges, I took great interest in all things oxytocin. So I'll share just a little bit of my personal background information. I completed my undergraduate degree at U of T a few years ago. I majored in health and disease under the human biology department and did a double minor in psychology and French. Took some time to travel after I completed my degree, and then upon returning to Toronto, I've enrolled in a psychology certificate at Ryerson University and am now pursuing a career in clinical psychology. So I'm essentially extending that psych minor from my undergrad at U of T in preparation for graduate studies in clinical psych. So while taking these courses at Ryerson, I took on a research assistant role at the Science of Music, Auditory Research and Technology Lab, or the SMART Lab. While at the lab, I've spent much of my time assisting on the Singwell Initiative, which ultimately served as the basis of the research paper. So Singwell is a project that is looking at the effects of group singing on social well-being, specifically for older adults with communication disorders, such as deafness, hearing impairment, or aphasia. The project began by looking at subjective measures of social well-being, such as happiness questionnaires or levels of social interaction. It has now evolved into looking at biopsychosocial measures, such as levels of cortisol and oxytocin in response to singing in a choir. So here I will show you some results from those biopsychosocial measures that have been collected and analyzed from saliva samples. Now we can see here on the left-hand side that results indicate that group singing has the potential to lower cortisol, and looking to the right, increase oxytocin levels as per measured in the saliva. So in other words, singing in a choir may enact quantifiable reductions in stress and enhance social bonding. So the Singwell project essentially acted as the starting point of my paper, in which I looked to explore oxytocin in greater depth. So the approach I took towards my paper was essentially a broad sweep of existing oxytocin literature in order to examine the feasibility of its therapeutic use. Additionally, I honed in on the potential to boost oxytocin through activities such as singing, so as to apply my paper to the Singwell project. Now, I began by searching for literature across the 5 databases shown here: Science Direct, Scholars Portal, PsycInfo, PsycArticles, and the online Ryerson Library database. I use search items oxytocin plus social bonding or oxytocin plus social behavior. Now, from all studies retrieved from the preliminary search, titles and abstracts were reviewed to determine suitability for inclusion in the review. As I was only interested in oxytocin as a therapeutic agent for humans, studies involving animal subjects were excluded, as were studies that were not peer-reviewed and those published longer than 15 years ago. Otherwise, no exclusions were made based on demographic data such as participant age or physical health, psychological condition, or study design. Now, of the 61 articles initially selected for review, 16 articles were excluded based on the criteria I mentioned. 30 original research studies were included in the analysis, as well as 14 review articles, which were incorporated for background knowledge and reference. In addition to the acquired peer-reviewed literature, a press release from biotechnology company Salimetrics, which outlined a novel salivary oxytocin measurement tool, was included in the review, and I will get into more details about that in the next few slides. So upon review, selected articles from all searches were organized into 5 thematic categories: methodological challenges in oxytocin assays, physiological intricacies of the human oxytocinergic system, interpretation of intranasal oxytocin studies, contribution of individual traits and contextual circumstances to oxytocinergic effects, and choral singing and oxytocin. Now I'll go through each of the themes in more detail. So the first theme we will discuss outlines the methodological challenges in the oxytocin literature. That is to say, problems revolving around the actual measurement of oxytocin. Now, to provide some context, oxytocin levels can be measured by assay, which, broadly speaking, is a laboratory procedure used to indicate the presence and quantity of an analyte within a sample. Historically, oxytocin activity has been measured using various assays, namely radioimmunoassay, enzyme-linked immunosorbent assay, and electrochemiluminescence. Now, the mechanisms are fairly complex, so I won't get into a ton of technical detail, but essentially each method involves detecting desired analytes, oxytocin in our case, within a sample. This is done using a combination of antibodies, immunoreactions, and biosensors. So using these various assay methods, oxytocin has been detected and measured in different mediums or samples like plasma, cerebrospinal fluid, as well as saliva. Okay, now each specific assay method has its own advantages and challenges. For example, radioimmunoassay has high sensitivity, which is beneficial for binding analytes in low quantities. Say if you had just a tiny bit of oxytocin present in a saliva sample, RIA will be able to detect that. But this becomes problematic when examining samples with the potential of high interference. This is especially an issue for plasma, which will contain many different proteins as well as partial oxytocin molecules, so you may not get an entirely accurate reading. Other assay methods are time-consuming or are expensive because they can only be analyzed in commercial labs. So each method has its associated challenges that do need to be considered. There are also some more general methodological hurdles that the scientific community needs to overcome. So although we've been using assays to measure oxytocin levels since the 1960s, there's a glaring lack of validation and replication between measurements performed by individual research groups. Oxytocin levels from comparable studies have been reported as differing by upwards of 2 orders of magnitude. So this makes comparing results from separate studies nearly impossible. Even further, the need for a standardized measurement protocol is demonstrated by one major study, which compared cerebrospinal fluid and plasma samples collected simultaneously from the same patients undergoing brain surgery. So during the surgery, cerebrospinal fluid and plasma were collected at exactly the same time, and then oxytocin levels were measured using each of the 3 different assay methods. The authors compared the oxytocin measures from the 3 assay methods that I mentioned previously, and they found no correlation between any of them. So this study presents the fundamental measurement obstacle that must be overcome in order to clarify past results, as well as to produce viable future research. Now, I had mentioned that I included a press release outlining a novel salivary oxytocin measure by a company called Salometrics. So Salometrics is a reputable manufacturer specializing in salivary assays and has been considered a front runner in salivary science since 1998. So in this press release, the company presented specifications of a highly sensitive salivary oxytocin assay that has been extensively validated for sensitivity, precision, and specificity, and this has been done across laboratories. So although measurement methods in oxytocin research may be a little bit behind schedule, we are beginning to see scientists address this lack of validated measure. So this will hopefully lead to standardized protocol. Now, moving forward onto our next theme, we have the physiological complexity and intricacies of the human oxytocinergic system, as well as the challenges which this vastly complex system may present. So though we've begun to understand some of the physiological mechanisms underlying oxytocin's effects, the interaction between this system and other hormonal and neurochemical systems is still largely unclarified. Now, where this holds particular significance is between the sexes. So researchers have called attention to the potential safety concerns of including female participants in these experiments due to this unknown interaction with estrogen. But this, of course, results in a marked male subject bias in the oxytocin literature. So that is unrepresentative of the whole population. Now, a second challenge under this theme is the apparent fluctuation of the oxytocin system throughout the lifespan. Oxytocin research has focused largely on clinically healthy young adults, and longitudinal study is required, as some research indicates that age-related changes in the oxytocin system may invoke changes in social behavior later in life. Finally, oxytocin appears to have distinct yet interconnected roles in the central nervous system and peripheral tissues. Centrally acting oxytocin is signaled for release from a different region of the hypothalamus than oxytocin intended for peripheral tissues. So there's a spatial difference, and there also appears to be a temporal one, such that peripheral release is an instantaneous response to stimulation, but central release peaks several minutes after stimulation. So again, we've not yet clarified this exact interplay between oxytocinergic systems, and this avenue of research is really critical to our understanding of the entire system, as well as oxytocin's potential as a therapeutic agent. Now, on to our next theme, which is the interpretation of intranasal oxytocin studies. Now, when I say there's potential to use oxytocin as a therapeutic agent, there are essentially 2 ways in which this can be accomplished. So you can either participate in activities that seem to increase endogenous, that is our own oxytocin, like the Singwall Project, for example, or you can administer exogenous oxytocin that is added into our system, either through the nasal passage or through the bloodstream. Intranasal studies have provided critical contributions to furthering the field of oxytocin research. We've seen tons of experiments using intranasal oxytocin to look at changes in social behavior, changes in eye gaze, interpersonal cooperation. There's even been an increase in web searches for things like oxytocin autism nasal spray. And these searches have increased by more than 5,000% since 2004. So while the prospect of an oxytocin nasal spray is illuminating and hopeful, these studies must be interpreted with caution before we go any further. So firstly, there is no established optimal dosage for intranasal oxytocin. And considering the vast physiological complexities that I mentioned in the previous slide, this is a really critical point. Further, if intranasal oxytocin is meant to be available for commercial purchase someday, researchers must make certain how much oxytocin is both optimal as well as is safe. For example, one study found that in a small sample of males with autism spectrum disorder, 8 units of intranasal oxytocin increased emotion recognition of both happy and angry faces, but there was no significant increase in the effect after 24 units were administered. In other words, 8 units of oxytocin was enough to observe this effect in emotion recognition, but upping that dosage to 24 units made no significant difference. So while there's promise for oxytocin to be delivered this way, researchers must work on figuring out these exact dosages. Now, when considering intranasal oxytocin, we also have to look at the 3 distinct nasal pathways to the brain and the periphery. So when a nasal spray enters the nose, the spray may be absorbed via 3 different and distinct pathways, depending on whether the spray hits the front, middle, or back of the nasal cavity. Research has found that the 3 pathways have very different targets in the body. The first travels to the periphery through the nasal vasculature and the bloodstream, then to those peripheral tissues like the heart, the kidney, the mammary gland. The second pathway reaches the cerebrospinal fluid by way of sensory neurons and the olfactory bulb. Then through the cerebrospinal fluid, it reaches targets such as the limbic system and the prefrontal cortex. The final pathway takes the trigeminal nerve and reaches structures in the brainstem. Now, these delivery targets have potentially variable behavioral implications, such that oxytocin acting on peripheral tissues may exert a different behavioral effect than oxytocin acting, let's say, on the prefrontal cortex. The significance here is that we first need to clarify whether any behavioral differences do exist between oxytocin reaching these distinct targets, and then researchers will still need to devise a way to send that nasal spray towards a desired pathway. So even further than that, the unknown interplay between the central and peripheral oxytocin systems that I mentioned previously may also be at play here. So this is another fairly complex avenue that requires further exploration before oxytocin research can progress. Now, our second to last theme is the contribution of individual traits and contextual circumstances to oxytocinergic effects. This is a relatively novel discussion within oxytocin literature, as researchers initially believed oxytocin exerted effects in a situation stable or invariant way. And they also believed that these effects were generalized across all individuals. In other words, it was believed that oxytocin increased social bonding in everyone in any situation. We're now beginning to see that this may not be entirely accurate. I'll illustrate this point by comparing 2 studies. First, we'll turn our attention to the graph on the left-hand side. This is from a study by Domes and colleagues in 2007, which looked at the effects of a single dose of intranasal oxytocin on emotion recognition in a sample of 30 healthy male volunteers. On our y-axis, we have the percentage of correct responses in the task. So here, we can see the responses on all items. The white bar indicates the placebo condition, and the black bar represents responses after administration of intranasal oxytocin. We can see that oxytocin improved overall performance in the task compared with placebo. Now next, the authors looked at responses based on task item difficulty, easy versus difficult. Now here, we see no effect was detected on the easy items in the task, but here we see oxytocin improved performance on difficult items. Now, in 2010, Guastella and colleagues repeated this experiment, but this time with a sample of 16 male adolescents with autism spectrum disorder. Now we'll be turning our attention to the graph on the right. Now, like the previous study, we can see here that oxytocin improved performance on the task in comparison with placebo when looking at total overall responses. However, here we see an opposite trend when the authors divided the items based on difficulty. With oxytocin, this sample improved on the easier items, but there was no effect on the difficult items. So this is a really key comparison. At a first glance, the results look contradictory, but these seemingly opposing results can be resolved by this novel thinking that oxytocin has enhanced effects for certain individuals only in certain circumstances, rather than acting uniformly across individuals and situations. So perhaps this difference in oxytocin activity between these samples lies in the specific contextual or situational circumstances. That is, the difficult versus the easy task. And these effects also depend on who is performing those tasks, whether that be a typically developing individual or a person with autism. Further research needs to investigate such individual traits and situations, as well as differences in perception and motivation and even genetics when considering oxytocin as a potential therapeutic agent. Okay, now on to our last theme, choral singing and oxytocin. Biopsychosocial investigation indicates that group singing has several positive health benefits, including increased social well-being and increased social bonding. Now, when it comes to oxytocin activity, the few reported studies have shown inconsistent results. For example, in 2014, Günter Krutz looked to quantify changes in salivary oxytocin levels in a group of choir singers with varying degrees of singing experience. The authors compared oxytocin levels before and after choir session to those in a second experimental condition with the same participants. In this condition, they chatted in pairs about a recent positive life experience. The authors found no significant change in the oxytocin after the chatting condition, but salivary oxytocin did significantly increase after the singing condition. Now, on the other hand, Schlatt and colleagues in 2017 investigated changes in salivary oxytocin levels in experienced student singers in response to group singing versus solo singing. Now, in contrast to the previously mentioned results, the authors found a significant decrease in oxytocin levels after choir singing, along with a non-significant increase after solo singing. So these results seem to be opposing, but it is critical to consider these themes that I've outlined in this presentation when interpreting such results in the oxytocin literature. Perhaps there are individual or contextual differences between samples that may be at play here, or maybe there were methodological inconsistencies between measurement methods in the actual study protocol. So it will be absolutely imperative to address such challenges in order to clarify future oxytocin research when it comes to activities such as choral singing. So while literature investigating oxytocin levels in response to choir singing is for now pretty scant, the body of research is certainly gaining popularity, including, of course, the work being done at the Singwell Initiative. So extending this work does have implications for choral singing as a clinical intervention for both healthy individuals as well as clinical populations. Okay, now let us briefly sum everything up because I know I've given you a lot of information. First and foremost, scientists and researchers must validate and replicate adequate assay methods in order to standardize past, present, and future oxytocin research. Once appropriate measurement methods have been established, researchers have the possibility to clarify and hone in on those intricacies of the physiological oxytocinergic system. Oxytocin research can be significantly enhanced by defining protocol for exogenously administered intranasal oxytocin studies, and also by considering contextual and individual influences on oxytocin activity, especially throughout the lifespan. Finally, once all of these components are put into place, the potential of oxytocin as a therapeutic agent may be propelled from the confines of research laboratories to dynamic contexts for a wide variety of individuals. Okay, so that concludes my presentation. Thank you so much for your attention. Now we can move on to any questions you may have. I'll keep this slide up in case if anyone would like to use it as reference, or otherwise I'm happy to jump back to different points of the presentation if that helps.
All right, so first of all, let's all give Claudia a round of applause. That was a great presentation.
Thank you, thank you.
Yeah, so I'm going to start off with a question from Isha. In terms of the choral singing studies, do you think that the familiarity / relationships between singers could have been a confounding variable that could have contributed to the change in oxytocin levels?
Absolutely. Absolutely, like 100%. And it also depends too. So for example, with the Singwell project, we've been taking salivary measures from day one. So we take a baseline measure and then we're taking pre-post at let's say 3 weeks after choir singing, 6 weeks after choir singing. So most of these studies, it is sort of a longitudinal sort of framework essentially. So obviously you're not basing this off of one measurement, but absolutely. And like things like that do need to be considered in these studies. You know, that's exact. Thank you for that question, because that means you got exactly the point of the presentation. There's a lot of sort of different variables that do need to be considered when looking at those oxytocin levels. So absolutely, yes.
So just to clarify, have you, so when they start with Singwell, is it are they just being introduced as strangers?
Yeah, exactly,
Okay. And
exactly.
then do you notice anything as time goes on?
Um, it depends. Yeah, I mean, what do you mean? Like, what do I know? In what sense? Notice what?
Uh,
Like relationships
I
or?
meant, yeah, in terms of the oxytocin levels or any other measurements that you might take, because if I'm interpreting you correctly, then it looks like you take a measurement after like every few weeks or something.
So we're looking at pre and post the
Mhm.
choir session. So we take the salivary sample right before the choir session, and then we take it right after. So that's essentially what we're comparing. It's like a pre-post design.
Sorry, I guess what I'm asking is, do you notice a change in the difference over time as the choir continues to work together?
Yeah, yeah. It depends on the individual, to be honest. It depends, and that's kind of why I was stressing the individual differences because there isn't really a consistent result. I mean, overall, we are seeing that, yeah, we are seeing these increases in oxytocin as well as the decreases in cortisol, that first graph that I showed you. So overall, we are seeing that effect. But again, when you look at the individual people, it is variable. It is variable. So you can't necessarily say that they are overall increasing by XYZ. It is variable. And again, you have to consider, what sort of day that person had before they come, before they come into the choir session. There's so many variables that are kind of playing into it. But overall, we are seeing an increase. Overall, we
Mhm.
are seeing an increase. But when it comes to individually, then it becomes a little bit more complex. And
Of course,
that's
yeah.
sort of what I would like to get into more is like those individual things.
Makes sense, yeah. Jesse also has a question. Do you think that the results of choral singing would extend to other group music activities such as orchestras or bands playing together?
That's a great question. That's a really good question. Now, we focus specifically on singing because singing has been singing and musical instruments to an extent as well, but singing has such a long history with humans. We've been singing for thousands of years. It is sort of a thing that does bond people. And so that's sort of where all of this research stemmed from, as we see the health benefits of that. So essentially, as I mentioned again, the project started off by, let's get these people together. Let's see. And this is also why it started with this population, because older individuals do have a tendency they have greater social challenges, and loneliness becomes a factor, and other social sort of challenges that they come across. So we started off by looking at questionnaires. How do you feel? How are you interacting with people within the choir? How are you interacting with people outside of the choir? Again, before and after the choir session. And now we're starting to look at those biological measures. So as opposed to a subjective sort of questionnaire, we're looking at tangible, quantifiable changes in the biology of it, essentially. So we did have one condition. This was before I was part of the project, but we did have a condition that looked at yoga sessions, and there was a little bit of an oxytocin increase as well as the cortisol decrease, but not nearly as much as with the singing. So
Mhm.
and we're actually extending this project now as well. Obviously, now during COVID, everything's virtual. So we've extended the choir to a virtual choir for all individuals with mental health challenges. So we're kind of looking at different populations now, given COVID. Again, people are feeling lonely. People need more and more social interactions. So we're trying to figure out a way to get saliva measures with people doing it at home, which is a little bit complicated, obviously, considering COVID and saliva and everything as well. But we are trying. So hold tight. Hopefully, we'll get some more results for you.
Hopefully.
Yeah.
I imagine it would also be different depending on how the rehearsal is run. If it's a, okay, shut up while I work with this section of the choir versus a more
Like a group
I can
singing.
understand. Yeah.
Exactly. Exactly.
Um, yeah, so we also have a question about do you know if there's been research done into non-person or even non-in-person social bonding and oxytocin?
Like in humans, do you mean, or?
I'm assuming it's they're referring actually. I'm actually not sure. So let's assume both.
Can you run that question by me one more time?
Yeah, sure. Has there been research into non-person social bonding and oxytocin?
Okay, so non-person. So I'm gonna interpret that as like animal studies.
Oh, sorry. Non-in-person. Sorry, non-in like bonding that's like I'm guessing like virtual and of
Like
some sort.
like the virtual, yeah. So
Yeah.
not that I'm aware of really yet. I would imagine, probably in the next year or two, once publications start coming out, given COVID and everything is virtual, I'm sure so many people are seeing, there's tons of research projects that are going entirely virtual now. So I'm sure in the next year, I don't know, year or so, we'll see more. Not that I'm aware of just yet, but as I said, with this virtual choir that we are starting, we are looking to contribute to that body of research, because I mean, it's going to be really imperative to see what we can, because who knows how long these pandemic measures will go on for. Who knows, we're going to need to really put into place things that people can feel bonded with others. We need social interactions. We're social beings. That that's why we have these neurochemicals that respond to social interaction. I mean, it's literally in our biology. So I'm sure, I'm not aware of a ton of studies yet, but I'm sure that they will be coming out.
All right. Thank you so much, Claudia, for presenting.
Thank
It
you.
was great.
Thank
Uh,
you. Thank you so much, guys. Thank you.
yeah. All right. We're now going to take a break and come back at about 4:05. So.